All right, back again for round two. Woo! Okay, so now we're talking about the defense system. And by defense, so remember for this whole class, we're talking about form and function, and we're talking about those 7 different key aspects of life that all organisms have to engage in to survive. So there's different kinds of defense, but in this case, we're primarily focusing on the immune system and how it can defend against pathogens making you sick and taking you out. So first, we're going to talk about the innate immune responses that defend animals against broad groups of pathogens. And then we're going to take you through various aspects of the immune system in animals. So remember that there are 3 key processes that lead to success with immune systems. So the first step is to prevent the entry of potential pathogens. So the first part of your reading guide talks about some of those barriers to pathogens. So we're not going to cover that in any kind of great detail. Those are pretty straightforward, but we'll talk about them in lecture. So preventing entry is the first response. Detecting the presence of a pathogen by distinguishing it from the animal's own body. So it'd be really bad if your immune system couldn't distinguish self from not self, right? So it's got to be able to distinguish a pathogen. And then you have to eliminate the pathogen. So we're going to walk through especially points 2 and 3. So it's important to recognize, by the way, the difference between the innate immune response and the adaptive immune response, which we're also going to discuss. So an innate immune response is one that you're born with. You have it naturally. The adaptive one is one that changes as it's exposed to pathogens and adapts to those pathogens. So we'll talk about that part in a minute. So with respect to the innate immune response, you have several different cells that collectively respond to pathogens. And so these are white cells primarily, so leukocytes. And you can see several different examples here in these sort of stylized diagrams. So monocytes, eosinophils, lymphocytes, neutrophils, and basophils. So these cells collectively will mount a response to pathogens. And they can distinguish between different groups of pathogens, so bacterial or fungal or viral, but not specific strains within groups. So these guys sort of mount a generic response. So how does that work? So let's talk about some of those details. Now remember, most of the stuff that we're talking about in this class is probably review from stuff that you've had in other classes. We're trying to get you to think collectively about different sorts of organisms. So how animals react, how plants react, and how others respond to certain situations. Okay, coming back to this. So you have these things in your body that are referred to as toll-like receptors. So these are a subset of a larger group known as pattern recognition receptors. And by the way, they're called toll-like receptors because there are these toll genes that were discovered in the 80s. And without going into a long digression, these particular proteins have DNA sequences that are very similar to those toll genes. And so that's why they're called toll-like receptors. Anyway, so what they do is these proteins act as a receptor that receives the molecular signal that a pathogen is present. So this is one of the ways that your body recognizes pathogen from self. Okay? So when it recognizes that a pathogen is present, right, it's recognizing an antigen. There we go. Now, 11 different TLRs have been identified in humans, each responding to one or more antigens. And you can see them arrayed here in this slide. So you have some fungal antigens that are responded to by these proteins. You have bacterial strains. You have viral strains. Motile bacteria versus gram-negative bacteria. So it's just a diagram showing you the different kinds of antigens that at least TLRs in humans respond to. So how do these work? So the antigen binds to the TLR on a leukocyte, which is the site's going to be on the plasma membrane. And what this will do is this will kick off or signal a cascade in the cell that initiates the appropriate immune response for that pathogen. So this is sort of a generic, here's what happens. So the pathogen, the antigen attaches to this protein on a membrane, and that's going to signal a series of reactions that are going to respond to that pathogen. So we're going to talk about how those responses work. I feel like my arrow's not working. Okay, so let's dig down into a little bit more detail. So what are the components of the innate human response? So you have several different cells. You have macrophages. So these are going to kill invading cells via phagocytosis, but they're also going to release cytokines. And these cytokines are really important because they basically send out a signal telling other immune cells to come here and fix this problem. There's a pathogen here. It's kind of like using a blowhorn to say like, hey, we got pathogens here. Help me, help me, help me, help me. And so it sends out this chemical message that recruits cells to come help. Neutrophils are also going to kill pathogens via phagocytosis, and these guys are just like the Pac-Man, like they're going to eat up all the pathogens. And then there's mast cells. So these guys are going to help by increasing blood flow to an infected area. So let me show you how this works, but just to reiterate, so this is a good thing because you want to get more blood into the area because that's going to help these the recruitment of these helper cells to fight off whatever is the pathogen that's invaded the system. So here's kind of what's happening. So imagine that you've got some sort of an injury right here, like you've cut yourself and you're starting to bleed. So this is your skin out here, and then here's your circulatory system. Here's those red blood cells that are coursing through that vessel, and you can even see these yellow platelets here. So of course, the first part of the response, right, is injury reparation. So you're going to have platelets coming in here that are going to try and, basically clot up the injury to prevent you from bleeding. But then these blued sort of lozenge or pill-shaped forms, this is supposed to be some sort of pathogen. So maybe there's some sort of thing on your skin, bacteria, could be anything. And so remember that, the first part of prevention is to try to prevent the pathogen from getting in your body in the first place. And one of the easiest way for pathogens to get in is through a wound. So here's this wound, here's this pathogen, right? So it's going to enter into the wound right here. All right, the platelets are going to release blood clotting proteins, which we already said. So it's trying to clot it up to prevent pathogens from coming in the first place. But at the very beginning, could be really difficult in a particular situation, they get in. So the injured tissues are going to release cytokines, recruiting immune system cells. So here's these chemokines that you see going out. And remember, macrophage, so anyway, these cytokines go out basically saying, help, help, we need the helper cells to come help get rid of these pathogens. So mast cells are going to start first. So they're going to secrete histamines. That's going to dilate your blood vessels, allowing more cells to come into the system. The recruited neutrophils and macrophages are going to remove pathogens by phagocytosis. So they're going through and they're going to eat them all up, right? Hopefully effectively. And then just to kind of show you this happening, there's a cool video. There's no audio, but check it out. So this is what you're going to focus on, right? So here's a macrophage, and it's going to chase these little jerk pathogens. Check this out. I don't know what this thing hanging on his tail is back here, by the way, but it's going after these two guys, these two little jerk pathogens. Okay. Now they're giving him a run for his money because they're running. They think they're going to get away. This dude, you can smell the failure about to happen. Here it comes. They're not getting away, and then boom! And you're done. Pathogen neutralized. Okay. So that's a gross overview of the innate system, and we're going to go in more details in just a minute. But now let's talk a little bit about the adaptive immune response. So generally speaking, the innate response initially is always going to be faster than the first time a pathogen enters your body that your body's not used to, so the adaptive response would kick in. So the first time a new or novel pathogen enters your body, the adaptive response is going to be slow. But once you've developed antibodies towards it, then it's always going to be faster than your innate response. So let's talk about how adaptive works. So the first step is, again, recognizing when a pathogen is a pathogen. So there's basically 4 key features to the adaptive response. The first is specificity. So antibodies and other components should bind only to specific sites on specific antigens in order for this to be a really effective immune response. Second part is diversity, though. So hopefully it's going to recognize virtually any type of antigen. So it has both specificity and diversity at the same time to be truly effective. There's got to be memory to it, right? So responses are stronger and quicker when an individual is exposed to antigens from previous infections. So think common cold, or maybe a better one would be the flu, right? The more times you get exposed, you're going to heal much more quickly. And then non-self recognition. It's always key, right? So molecules that are produced by an individual do not normally trigger a response. Okay. So who's all involved in this? So lymphocytes are your major players here. So the cells that are involved in the adaptive immune response are these lymphocytes. Now, these are, we're only going to cover 2 types of cells, the 2 main players. So these are cells that are going to be produced in your bone marrow. So both T cells and B cells are going to be produced, or lymphocytes and T lymphocytes and B lymphocytes. Now, they're both produced in the bone marrow, but T cells are going to mature in your thymus, whereas your B lymphocytes or your B cells are going to mature in bone marrow, hence T cells versus B cells, right? Now, once they've matured, they're going to be transported through your lymphatic ducts and your blood vessels, and you're going to see that there's a lot of activation in the spleen and then in the lymph nodes found throughout your body. So sometimes, just as an aside, sometimes you might notice before you even get sick, you might notice that something is happening. Like you can actually feel your like you have clusters of lymphocytes, say in your armpits and your groin area. If you feel swellings or soreness there, that's usually saying that your body, so that's your body responding to some sort of pathogen. Okay, so T cells, B cells, how do these things work? Let's step through this. So T cell receptors, or TCRs, so these are proteins that do not bind antigens directly. Okay, B cells do, but T cells do not. So what'll happen is that helper cells will present pieces of antigen to the T cell. And this is sort of a stylized diagram. This is how T cells are generally represented. So this is the membrane of the T cell itself. This is the antigen binding site, and there's 2 chains. There's an alpha chain and a beta chain. Contrast this with the way B cells look. So B cells are receptor proteins. So there's the same shape as antibodies produced by the B cells. So, and they are going to bind directly. So here's the membrane of a B cell. Here's the antibody here. So notice, I'm going to back up. So here's the antibody for a T cell, sort of straight up and down. A B cell has this sort of Y shape, and it includes heavy chains as well as light chains. And the antigens are going to bind here on the ends of the Y. Okay, so you got B cells, you got T cells. How do they interact? Okay, and again, we're talking about the adaptive immune response. Okay, so here's an antibody, and this is a typical sort of B antibody, right? So antibodies are proteins that bind to a specific part of a specific antigen. Okay, produced by the B cells, and their shape-specific binding. So they're generally speaking, you find in slides where they're trying to depict what's happening, they'll show you different antigens. Where'd my cursor go? Different antigens as different shapes and different colors, right? Because they're very specific. So think of these as being the pathogen or part of the pathogen that's being introduced into the body, and they're going to attach to these binding sites on these antibodies, which are on your immune cells. Okay? All right, stick with me here. Okay, So let's go into more details. Let's compare and contrast the steps for activating B cells and T cells. So what happens when a pathogen enters your body? So the clonal selection theory goes like this. So here you have all kinds of B cells, right? And notice they've got these antibodies attached to them. Okay, and notice that they're different colors. So these are going to represent different kinds of antibodies, and these are antibodies to certain kinds of pathogens. Okay, so antigens recognized by receptors on specific lymphocytes. So let's say you've got this particular pathogen, and presumably it's entered your body before. That's why you have antibodies to it. And they're going to bind to these antibodies, these receptors on these B cells, right? And here you go. Here's a bunch of them responding, or attaching rather. When this happens, and when there's a critical threshold for them, when enough of them have passed that critical threshold and entered into your body, the cross-linked receptors are going to activate these lymphocytes, right? So basically it's going to go into the nucleus, and it's going to tell it to start cloning itself. So it's going to create lots more clones of itself to help attack this pathogen that's entered your body. So matching lymphocytes are cloned. And then what happens? So with T cell activation, what happens is this, let's say this antigen comes in contact with the cell. It's taken into the cell. It's basically broken up into pieces. Oop, hold on, I forgot to do this. Here we go. So these are dendritic cells. So you need their help when it comes to T cell activation. So dendritic cells are going to ingest antigens by phagocytosis. So they're going to take it in. And when they do, they're going to break apart this antigen. So there's enzymes inside these dendritic cells that are going to, basically cut it up. And then little pieces of that cut up antigen are going to be bound to or loaded onto these MHC proteins. So major histocompatibility proteins. So you see that little bit going on right here. Okay. So when that happens, they're then going to be they're loaded onto the MHPs, and then these MHPs are going to be transported to the surface of the cell, where they're then going to hang out right here at the surface. So here they are with a little piece of that antigen. Okay, and watch what happens. So here's this dendritic cell with that MHC complex holding on to that a little bit of that antigen. Okay. Now what's going to happen is the T cell receptor binds to presented antigen fragments, and T cell is activated. So here you have this is the dendritic cell, right? And here you have two different kinds of T cells, right? So they're going to bind to these T cells. That's going to activate them, and you get two different kinds of T cells that are going to get activated. You have helper T cells, and then you have cytotoxic T cells. So the T cells are going to differentiate into these two different types. And then what happens is these helper T cells are going to release cytokines, recruit more cells, and they're going to help to create and activate B cells and T cells, but cytotoxic T cells. When they activate those cytotoxic T cells, these T cells are then going to kill all those pathogenic cells that are inside the cell. Right, so they're going to kill off all the pathogens within that cell. And then remember, we said they're going to activate B cells too. So what's happening with the B cells? So here you have the antigen binding to the B cell receptor. It's going to do a similar thing. So antigens are going to be taken in. They're going to be cut up by enzymes. Those cut up enzymes are going to attach to the MHC, which are then going to come to the surface of the cell. And you see them here, just like you did with those T cells, right? Now this happens to be a class 2 type MHC protein, but we're not going to go into the class 2 versus the different other classes. In any case, so they then present on the surface. They then are going to bind to these helper T cells. Cytokines are going to be released. Sorry, I forgot to pull up my little text here. So the B cell is going to stimulate a helper T cell. Right? Cytokines from the helper T are going to be stimulated, so it's going to recruit more cells, and it's going to activate the B cell. So it's going to activate the B cell and cause them to replicate. Okay, so they're going to clone themselves. They're going to some will differentiate into plasma cells, which produce antibodies. And here, what's key is that these antibodies don't necessarily need to be attached to cells. They can actually float around in the system, and whenever they encounter a pathogen, they can attack that pathogen directly. So yeah, antibodies will bind to antigens and mark them for destruction. Okay, I feel like this is a lot. I feel like this is a lot of moving parts, but stay with me here. And we're going to talk about this more in class too. So you can have a situation here called somatic hypermutation. So the DNA sequences in B cells that code for those antibodies can undergo rapid mutations that modify the variable region of the receptors. So that could be good and it could be bad. And we're going to talk about some case studies where that's true in class. So hold on to this information until we're in class. Okay, for the next learning objective. So now we're going to compare and contrast how the adaptive immune system responds to extracellular and intracellular pathogens. So there's two kinds of responses here, the humoral response and the cell-mediated response. The humoral response leads to the production of antibodies and other proteins secreted into blood and lymph, and it acts on the extracellular pathogens. Okay, the cell-mediated response is going to act on the intracellular. So let's talk about extracellular pathogens. So here's how this can work. So here you have some sort of a pathogen right here. So that's going to be the orange sort of lozenge or pill-shaped forms. And here's your antibodies. So when this pathogen enters into the system, now again, this is outside of the cell, what can happen then is you have these antibodies that will then bind to those pathogen. It's going to coat them, and that's going to basically send out a signal so that neutrophils can then phagocytose them and then destroy them. So that particular situation is called opsonization. So antibodies coat pathogens, helping phagocytes identify them and destroy them. So neutralization happens when coated pathogens, what happens there is you have coated pathogens. If they don't encounter a neutrophil and get destroyed in that way, if they happen to bounce onto a host cell, because the pathogens because the antibodies are coating it, it's blocked from infecting any kind of host cell, so they're neutralized. Now they're not destroyed, they're still out in extracellular space, but at least they're being blocked from harming any of the host cells. What will often happen then is as these float around, you'll have this agglutination or this basic clumping of pathogens together, which makes them even easier for neutrophils to find and phagocytose. And then you can have co-stimulation of complement proteins. So basically what's happening here is you have these antibodies that have attached to this pathogen, and they basically punch holes and start to destroy and tear apart these pathogens. So this is great. So when you have these extracellular pathogens, you can have opsonization happening, neutralization certainly, agglutination, which could lead to an easier opsonization, and then you can have co-stimulation of complement proteins. So that's all humoral immunity. So now let's talk, so remember that's all happening extracellular space. Now let's talk about the cell-mediated response, which acts on intracellular pathogens. So this is the activation of phagocytic cells and cytotoxic T lymphocytes, and occurs in cell-to-cell contact. So let's review this. So here's what's happening. So here's a cell, and it's gonna, let's say it's a virus-infected cell, right? And you can see what's happening here, right? You already have your MHC protein sitting out on top with a little piece of the antigen attached to it. So first, you have to have your cells recognize that that's a virus-infected cell. So the cytotoxic T cell recognizes and binds the infected cell. Okay, so here's your cytotoxic cell. It's going to bind. Here's the bound part. And this is a close-up of what's going to happen. So remember, now we're talking about what's happening in the intracellular space. So here's the infected cell, and then here's the part of the cytotoxic cell that's attached to it. What's going to happen after it's recognized it and bound to it? The T cell is going to secrete proteins that form pores and apoptosis activating proteins that enter the pathogens. So you can see here, So these green pore-forming proteins, they're basically going to break a gate through these membranes. And then these apoptosis activating proteins are going to enter into the infected cell. And basically, they're just going to punch holes into this infected cell. The infected cell breaks into fragments that are consumed. So it's going to do this. So after they've made these pores and then gotten the apoptosis activating proteins in there, it's basically going to torpedo. It's like setting a bomb off in that cell. It's going to break it into a bunch of bits. And then these macrophages are going to phagocytose them and eat them up. I mean, this immune system in your body is just like badass at like attacking these pathogens. There's a war going on in your body all the time. So just to give you a sense, so we've only barely scratched the surface of this topic, but there are over 100 different types of autoimmune disorders, as you can see here, and impacting various parts of your body, from your thyroid to your bones, muscles, skin, lungs, nerves, your GI tract, your brain, all different kinds. And your body is constantly responding to pathogens that enter your body and try to impact it. So for this last objective, we're going to compare and contrast primary and secondary immune responses. So in responding to future infections, especially when we're talking about the adaptive immune response, right? The primary immune response occurs the very first time an antigen is encountered, but there is a very definite way for your body to remember that antibody, and then to respond to it. So the secondary immune response is if the same antigen enters the body again. And of course, sometimes it's not exactly the same antigen entering the body, right? It could have been mutated, could have been altered. So how does your body respond to that? Okay, so activated B cells and T cells can also produce what are called memory cells. So memory cells remain in the spleen and in the lymph nodes for years or decades, and at any time could mount a rapid response to that pathogen should it enter your body again. So that's known as immunological memory. So how does that work? Well, we can actually stimulate that with vaccines. This is the critical importance of vaccines, as I'm sure all of you guys know. So a vaccine contains antigens from a pathogen or a killed or weakened version of the pathogen itself. So after vaccination, the body mounts a primary immune response that produces memory cells. And if a second infection occurs, then those memory cells are going to respond quickly to the threat. So I thought this was kind of funny. Keep calm and get vaccinated. All right, well, how do these vaccines work? So there's 3 different forms. You can get subunit vaccines, which consist of isolated viral proteins. Inactivated viruses that have been damaged by chemical treatments or exposure to ultraviolet light. So they do not cause infections, but are antigenic. Okay, so what that means is your body can still produce antibodies to it, but the pathogen itself has been inactivated. So it still, can mount a response, but you're not going to get sick from it. And attenuated or live viruses consist of the complete virus particles, but they've lost their ability to grow rapidly in normal host cells. So you're usually getting one of these 3 forms when you get a vaccine. So when it comes to things like flu or HIV, so these are pathogens that are extremely successful at mutating. There's a number of reasons for that you've probably already covered in cell class. But as a result, there's like an evolutionary arms race that your body hopefully is going to be able to wage, because each time you get exposed, like the likelihood of you being exposed to the very same flu virus that you may have been exposed to before is probably pretty slim. Now, that's not to say that many aspects of that virus are the same. It could only be a point mutation that has changed. But that could be enough to make the reaction of your body change. Okay, but most of the time it's pretty close. So memory cells that were effective during a previous infection by these viruses are unlikely to bind to the changed epitopes and trigger a response to a later infection. So as a result, it's extremely difficult to design an effective vaccine against these agents. But they can come close. Whoop, uh-oh. let me go back. So it's important to note, we're going to talk about some case studies in class, but it's really important to know that particularly when it comes to flu, studies have showed repeatedly that, oh wait, I don't want to tell you this because I want you to do a thought question in class. So never mind, skip that. Okay, that's all I got for you today. I'll make another video for the material for Thursday, and then we'll do some active learning in class. Enjoy!